On the left, we see the 5' end of the leading strand, the strand on top, with the 3' end on the right. The leading strand will always be the strand that runs in the 3' to 5' direction in relation to the direction of replication, as the DNA polymerase III is able to bind to the 3' end and replicate continuously. For the lagging strand, the strand on the bottom, we see the 3' end on the left and the 5' end on the right. The lagging strand, which is anti-parallel to the leading strand, will always run in the 5' to 3' direction in relation to the direction of replication. RNA primase is responsible for placing RNA primers along the DNA strand to give DNA polymerase III a site to bind to. The presence of RNA primers is necessary because they provide the 3' OH that DNA polymerase III is able to bind to in order to begin the replication process. Gyrase is a topoisomerase that is responsible for relieving the tension from the supercoiling of the DNA strand. This helps prepare the strands for further separation by helicase. Helicase is responsible for unwinding the DNA helix and does so by attaching to the replication fork and breaking the hydrogen bonds between the nucleotide bases. This action is necessary to provide separated strands for DNA polymerase III to replicate. Okazaki fragments are the fragmented pieces of DNA that are replicated on the lagging strand of DNA due to DNA polymerase III only being able to replicate in the 5' to 3' direction by adding to the 3' end of the existing DNA strand. Because the lagging strand is in the 3' to 5' direction, DNA polymerase III has to jump backward in order to replicate pieces of DNA in the correct direction. The function of DNA polymerase I is to remove the RNA primers and connect the Okazaki fragments that have been created on the lagging strand by the action of DNA polymerase III. This is necessary to fill in the gaps of the new strand that are missing DNA. Ligase is responsible for sealing the nicks which are created by the removal of RNA primers, which leaves a portion of the lagging strand without a sugar phosphate bond. DNA polymerase III is responsible for pairing complementary nucleotide bases to replicate the DNA template strand. Its role is essential in ensuring that the appropriate bases are paired together to create the new strand of DNA. Single-stranded binding proteins are responsible for keeping the two original DNA strands separated, as well as stabilizing them to allow for the replication process to occur. This is important to help prevent hairpins, or double-stranded DNA, from being formed. Phosphodiester bonds connect the 3' end of one nucleotide bond to the 5' end of another nucleotide bond, thus attaching the sugar phosphate backbone, which helps hold the DNA structure together. And finally, the origin of replication is the site at which DNA replication begins.